Welcome to Biology 101. Today we will be covering biological macromolecules. Firstly, you'll have a brief review of chemical concepts. Next, you'll learn how macromolecules are formed and decomposed. And then you will learn about the types of macromolecules, their structure, and their properties. Finally, we'll do a quick review of the concepts that you learned throughout this video. The atom is considered the basic building block of matter. It consists of subatomic particles, called protons, electrons, and neutrons. The nucleus is considered the central core of the atom, consisting of positively charged protons and neutral, no pun intended, neutrons. Generally, atoms with the same number of protons constitute an element. So in this case, we have 2 protons. So this is what we call a helium atom. However, if there are a different number of neutrons, then we have what we call a different isotope of that element. So let's say we took away one of the neutrons in helium. Let's take away this neutron. The resulting atom would have 2 protons and 1 neutron and 2 electrons, resulting in helium-3. We call it helium-3 because it has 2 protons and 1 neutron, and together that makes 3. The electrons also have a few unique properties. Electrons have a negative charge and are high in energy but have a negligible mass. They also travel around the nucleus at varying speeds and distances. Now the topic of electrons transitions nicely into electronegativity. Electronegativity is the tendency for an atom to attract electrons from other atoms. This is because most atoms are not stable in their natural state. So, they react with other atoms and electrons to become stable. Generally, we will see the electronegativity increase as we go up and to the right of the periodic table of elements. 3 elements that have a very high electronegativity and that we often see reacting are oxygen, nitrogen, and fluorine. Now I like to remember these elements using the abbreviation FON because it kind of sounds like fun. Now electronegativity plays a very large role in the sharing and transfer of electrons between atoms. And that leads us to intermolecular bonding. Intermolecular bonds are bonds that hold atoms together. And different types of bonds will form due to the difference in electronegativity between atoms as electrons are transferred and shared. A nonpolar covalent bond is a bond where electrons are shared equally. The difference in electronegativity between the 2 atoms will generally be less than 0.4. A polar covalent bond has an unequal sharing of electrons, resulting in partially positive and partially negative ends. Polar covalent bonds will have an electronegativity between 0.4 and 1.8. Finally, ionic bonds don't share electrons at all, and instead completely transfer electrons because of the high electronegativity difference between the 2 atoms. Ionic bonds form when the electronegativity difference is greater than 1.8. Now these bonds allow compounds and molecules to be formed. Another type of bonding is intermolecular bonding. Now intermolecular bonding occurs due to the electrostatic attractions that occur between molecules. And this happens due to the attraction between opposite charges. As we can see here, we have a water molecule. And we know that a water molecule is a polar molecule. Polar molecules have a partially positive end and a partially negative end. And we can see that due to the polarity of these water molecules, opposing charges are attracted to one another, forming what we call a hydrogen bond. Hydrogen bonds occur when molecules containing highly electronegative elements, including fluorine, oxygen, and nitrogen. Then again, there's our abbreviation that we had before, PHON, which associate with molecules containing hydrogen. So in this case, we have a water molecule which has oxygen and hydrogen. And due to the polarity of that bond, water molecules are able to form hydrogen bonds with other water molecules. Now, more specifically, water has an OH bond, which has a very big electronegativity difference between the two molecules, giving it a partially positive and a partially negative end. The polarity of water allows it to form hydrogen bonds, as we saw before, when associated with other water molecules or other polar molecules. And as we know from the previous video that we made, the polarity of water gives it some unique properties that are very important for the function of life. Another important compound is what we call an organic compound. Organic compounds are compounds which contain carbon atoms. Carbon is a central atom in an organic molecule because of its ability to form up to 4 covalent bonds with other atoms. And this happens because carbon has 4 valence electrons. Often, you'll see that carbon is linked covalently by sharing its valence electrons with the elements hydrogen, oxygen, and nitrogen. Now, these organic compounds will come in various shapes and sizes, including branched, chained, or ringed structures, including what we have here, which is pentane. Now, you may be wondering, where do organic compounds get their properties from? Well, the unique structural arrangement of atoms and bonds within the compound are responsible for determining the organic compound's properties. Now, the unique structural arrangement that we're talking about here is what we call the functional group. There are 7 different functional groups that you should generally know or have in the back of your head. First is the hydroxyl group, which contains an OH group and is polar and is often found in glucose and alcohol. Next, we have methyl, which contains a carbon atom bonded to 3 hydrogen atoms. Now once again, we can see how the tetravalence of carbon is going to be really beneficial, because carbon here is bonding to 3 hydrogens, and it's bonding to the rest of the molecule. Carbonyl functional groups occur when carbon is double bonded with oxygen, thus resulting in a polar functional group. Now, carbonyl is often found in things like acetone. Next, we have carboxyl groups, which occur when carbon is bonded by both an oxygen and a hydroxyl group. This functional group is charged and ionizes to release hydrogen ions in solutions, often making those solutions acidic. Next, we have the amino group, which is composed of a single nitrogen atom bonded to 2 hydrogen atoms. Amino groups are charged, accepts hydrogen ions to form ammonia when in a solution, which would then be considered basic. Next, we have the phosphate group. Phosphate groups are composed of a phosphorus atom bonded to 4 oxygen atoms with 3 single bonds and 1 double bond. Phosphate groups ionize to release hydrogen ions into solution, thus making them acidic. Finally, we have the sulfhydryl functional group, which is composed of sulfur atom bonded to a hydrogen atom. This functional group has the property of being polar. Now that we have some basic chemical knowledge of how compounds are formed, we can look a little bit more into how macromolecules are formed and decomposed. Most macromolecules are formed through a process known as dehydration synthesis. In dehydration synthesis, monomers combine to form polymers after water is removed. In this representation, we can see that a hydrogen atom and a hydroxyl group are removed from the monomers, thus forming water. The water molecule is removed, thus allowing the 2 monomers to bond together and form a polymer. Molecules are broken down in the opposite manner in a process known as hydrolysis. In hydrolysis, the polymer will break down into its constituent monomers after water is added. Now, more specifically, the water molecule is broken down into hydrogen, a hydrogen atom, and a hydroxide, thus going to the two monomers. Now that we know how macromolecules are formed and decomposed, we can go a little bit more in depth into what the macromolecules actually are. The first macromolecule that we'll be covering is a carbohydrate. Carbohydrates are sugars and polymers of sugars which have the function of storage and structure. Carbohydrates consist of monosaccharides, or simple sugars, which typically contain a carbon to hydrogen to oxygen atom ratio of 1 to 2 to 1. An example of this is glucose, which has 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms, as we can see here. And that follows the 1 to 2 to 1 ratio. These simple sugars are often linked together by covalent bonds called glycosidic linkages, which form through dehydration synthesis, thus forming a disaccharide or a polysaccharide. Specifically, a disaccharide forms if two monomers combine together. In this example, if we combine two glucose molecules by removing water, then we will form a maltose macromolecule. Some other examples of disaccharides include sucrose, which forms from fructose monomers, and lactose, which forms from galactose monomers. Polysaccharides form when two or more monomers combine. Some prominent examples of polysaccharides include starch, which is responsible for plant storage, glycogen, which is responsible for energy storage in animals, cellulose, which is responsible for the structural integrity of a plant's cell wall, and chitin, which is responsible for the exoskeleton of arthropods and the cell wall of fungi. The next macromolecule that we will be talking about is the lipid. Lipids are large nonpolar hydrophobic macromolecules that do not form polymers. Lipids have the function of providing structure in the cell membrane, serving as energy storage, and providing signaling molecules that allow the body to function. In biology, we will be separating lipids into three main categories. The first category is fats. Fats are composed of triglycerides, which are formed from glycerol molecules linking three fatty acids together. So here we can see the glycerol forming the three fatty acids, linking the three fatty acids together, thus forming a triglyceride. Now there are a few different types of fats, the first of which being saturated fats. Saturated fats have no double bonds in their hydrocarbon chains. As we can see here, there are no double bonds. As a result, saturated fats are often solid at room temperature and are often found in animal. So an example of this is butter. Butter is primarily composed of saturated fats, and we can see that because it's a saturated fat, it's solid at room temperature. The other type of fat is unsaturated fat. Unsaturated fat, unlike saturated fat, includes double bonds or triple bonds. As a result, unsaturated fats tend to be liquid at room temperature. An example of unsaturated fat that you may know is vegetable oil. And we know that it's an unsaturated fat because it's liquid at room temperature. The next type of lipid is what we call a phospholipid. Phospholipids are composed of a hydrophilic head, which is composed of a phosphate group, and a hydrophobic tail, which is composed of two fatty acids, one of which is saturated and the other being unsaturated. The unique duality of hydrophobic and hydrophilic properties of phospholipids allow them to play a vital role in the cell membrane, where they form what we call a lipid bilayer. The final type of lipid is what we call a steroid. Now, steroids are composed of 4 linked carbon rings and are often hydrophobic. So here we can see that this steroid has 1, 2, 3, 4 carbon rings. Examples of steroids include hormones such as estrogen and cholesterol, which plays a vital role in the fluidity of the cell membrane. Proteins are macromolecules composed of a central carbon atom linked to an amino group, a carboxyl group, and a unique side chain, which we will be calling the R group. Now, the R group is really important because they determine the structure of proteins. Generally, proteins have the function of structure, regulation, and the function of tissues and organs within our body. Now, proteins have complex levels of construction, resulting in structural levels. The first structure is the primary structure. The primary structure results from a sequence of amino acids linked by peptide bonds, thus forming a peptide chain. Now, the primary structure is indirectly determining the 3D structure of the protein. The next structure is the secondary structure. The secondary structure forms due to hydrogen bonds forming between neighboring peptide chains. As a result, they form shapes known as alpha helixes, as seen here and here, or beta pleated sheets, as we see here. We can see the hydrogen bonds being formed between peptide chains in the models that we see to the right. These attractions change the secondary structure of the protein. Next is the tertiary structure. The tertiary structure forms due to chemical interactions between the R groups, or the side chains, and directly determine the protein's 3D structure. Now, these chemical interactions between the side chains occur due to a variety of reasons, including hydrogen bonds formed due to opposing partial charges of the side chains, hydrophobic interactions occurring between molecules that have similar nonpolar tendencies, disulfide bridges, and ionic bonds. The final protein structure, and most complex, is what we call the quaternary structure. The quaternary structure forms when multiple peptide chains form together to form the final protein. Now, not all proteins will have this level of complexity, but we can generalize this as the final form of the protein's 3D structure. Problematically, proteins are very sensitive to environmental conditions. For instance, if the protein is subjected to high heat, acidity, or high ion concentration, it can result in what we call the denaturization of the protein. For instance, under high heat, the hydrogen bonds between the peptide chains could be broken, thus resulting in the tertiary structure, or the 3D structure of the protein, being broken down, resulting in an inactive form of the protein. The final macromolecule we'll be talking about is the nucleic acid. Nucleic acids are macromolecules composed of monomers called nucleotides. Nucleotides include a nitrogenous base, a phosphate group, and we can remember phosphate group as a functional group from before that we learned about, and a 5-carbon sugar. Nucleotides generally have the function of transmitting and expressing hereditary information. Now, there are 2 types of nucleic acids that you probably already know. The first of which is DNA. DNA, or deoxyribonucleic acid, includes a 5-carbon sugar called deoxyribose, nitrogenous bases, thymine, cytosine, guanine, and adenine, and a sugar phosphate backbone. Uniquely, DNA has a double-stranded helix shape. On the other hand, RNA, or ribonucleic acid, is composed of the 5-carbon sugar ribose, has the nitrogenous bases uracil, adenine, guanine, and cytosine, also has a sugar phosphate backbone, but, unlike DNA, is single-stranded. So what did we learn in this video? Well, first, we covered some basic chemistry on the formation of compounds, bonds, and how atoms interact with each other. Next, we discussed how macromolecules are synthesized and decomposed. Finally, we learned about the types and functions of macromolecules, which include carbohydrates, lipids, proteins, and nucleic acids, which all play an important role in the functions of living beings.